Very good afternoon to everyone present today. On behalf of Dr. Ashok K. Chauhan, Sir, Honorable Founder President, Amity Education and Research Group, our respected Honorable Chancellor, Dr. Atul Chauhan, Sir, and our dynamic Honorable Vice Chancellor, Professor Dr. Balvinder Shukla, Ma'am, I, Sudipti Kulshresht, PhD Research Scholar Bioinformatics, extend a very warm welcome to all the respected dignitaries, esteemed guests, deans, heads, faculty, students, and participants to the Skill Enhancement Workshop on Multi-Omics Data Integration and Data Analytics from 27th September to 30th September 2022. This workshop is being organized by Center for Computational Biology and Bioinformatics, Amity University, Uttar Pradesh, Noida campus, in collaboration with Ensemble Team, European Bioinformatics Institute or EMBL EBI, Cambridge, UK, Tauber Bioinformatics Research Center, University of Haifa, Israel, Pine Biotech USA, and Elucidata Corp, New Delhi. Let us start the inaugural session with the lighting of the ceremonial lamp. Thank you so much. To begin with, I would like to take the opportunity to invite Professor Kamal Rawal Sir, Professor and Head, Center for Computational Biology and Bioinformatics, Amity Institute of Biotechnology, Amity University, Noida. I would request Professor Kamal Rawal Sir to kindly deliver the inaugural address.
Thank you, Sudipti. I feel privileged and honored to welcome participants and colleagues to a very important skill enhancement workshop on multi-omics, data integration, and data analytics. During the next 3 days, we shall have presentation from our accomplished scientists from EMBL, United Kingdom, Haifa University, Israel, Pine Biotech, United States, Indiana University, USA, Elucidata, also from USA. Further, our MIT colleagues would be also sharing their research on last day of this workshop. This workshop is a part of our mission to impart skills to youth from biological domain so that they are able to face new set of challenges presented to us after COVID-19. Our institute, CCBB, Center CCBB, is on a mission to push the boundaries of knowledge and research. Through a unique research-powered education model, our students enrich their education through engaging in frontline faculty-led research. Our graduates are provided skills using our potent learning by doing system. As a center, we have obtained grants from DPT, ICMR, CERB, Ayush, Pfizer, ASCO, Kleber Foundation, and so on and so forth. We believe in collaborations and work with our partners from AIIMS, IIT Delhi, Rajiv Gandhi Cancer Institute, Baylor College of Medicine, Indiana University, and so on and so forth. Not only that, we are also commercializing our technologies. We are partnering with different companies. We are creating new use cases as well as launching our startups. In our center, we have initiated several new schemes, which include skill development, product building programs, and launch your own startups for students as well as for the faculty. We are laying emphasis on skill development along with the education. I believe to make this as a century of India, it is very important that youth of India should be equally proficient in education as well as skill. When it comes to the skills, your mantra should be skilling, reskilling, and upskilling. I'm sure you will move ahead at this pace, and with your skills, you will give direction to the better future of bioinformatics and biotechnology industry. On a technical side, high throughput next generation sequencing now makes it possible to generate a vast amount of multi-omics data for various applications. These data have revolutionized biomedical research by providing a more comprehensive understanding of biological systems and molecular mechanisms of disease development. Apart from that, the technologies such as deep learning algorithm have become one of the most promising methods in multi-omics data analysis because of their predictive performance and capability of capturing non-linear and hierarchical features in the data sets. We understand that multi-omics data have a role to improve prevention, early detection, and predictions. We can also monitor progression of disease, we can interpret patterns, and do endotypings. Using all these systems, we can design personalized treatments. Thus, in this workshop, we are looking forward to having a thought-provoking presentations and discussions which will contribute to our roadmap of multi-omics integration using technologies such as deep learning. Further, the workshops shall also offer a practical perspective into advantages, challenges, and barriers to implementation of new technologies in multi-omics data. I would like to conclude this address with the blessings of respected Founder President, Dr. Ashok K. Chauhan, Honorable Vice Chancellor, Dr. Balwinder Shukla, President STIF, Dr. Silva Murthy, and once again, my colleagues in CCVB. I would also like to welcome once again to this unique skill building workshop. Thank you very much.
Thank you, sir. Thank you, sir. I would now like to invite Dr. Priyanka Narad ma'am, Assistant Professor, Center for Computational Biology and Bioinformatics, Amity Institute of Biotechnology, Amity University, Noida, to give an overview of the workshop. Over to you, ma'am.
I hope I'm audible.
Yes, ma'am.
Okay. A very good evening to all of you, and I once again welcome all the participants to the workshop. To study complex biological processes holistically, it is imperative to take an integrative approach that combines multi-omics data to highlight the interrelationships of the involved biomolecules and their functions. With the advent of high throughput techniques and the availability of multi-omics data generated from a large set of samples, promising tools and methods have been developed for data integration and interpretation. Keeping all these in mind, this virtual work will focus on the use of public data resources and open access tools for enabling integrated working with an emphasis on data visualization. Working with public domain data can provide added value to data derived through a researcher's own work and additionally inform experimental design. This workshop is highly relevant in the current research scenario where an increased volume of data across the whole spectrum of biology has created both more opportunities and challenges to identify novel perspectives and answering questions in life sciences. The workshop will focus on issues around data integration and will include applications of machine learning approaches over the next 4 days. All participants kindly note that attending all the session and attempting quiz and feedback links is mandatory for successfully completing the workshop and the certificate. With these words, I once again welcome all the participants, our esteemed speakers from the industry and academia, and everybody to the workshop, and let us make it fruitful ahead. I hand it over to Sudipti to take it further.
Thank you, ma'am. I would now like to cordially invite Dr. Mohit Majumdar, Director, Global Business Development, Pine Biotech, USA. Dr. Mohit Majumdar has over 12 years of research and industry experience. He completed his master's in bioinformatics from Jamia Millia Islamia and a PhD in computational biology and chemistry from JNU. He is an expert in machine learning and application to biomedical and chemical data. He is currently heading the global business development for Pine Biotech, USA. and contributing to bioinformatics research and education by collaborating with universities in India and Africa, and mentoring participants from various programs by providing bioinformatics research support and applying statistical and algorithmic approaches. We are honored to have you here today, sir. I now request Dr. Mohit Mazumdar to address the audience and share his remarks.
Thank you so much, Sudipti, and thank you so much, Priyanka, and everyone who is organizing, who all the organizers, and also the members who have joined today. So in this very important skill enhancement workshop on a very advanced topic on multi-omics integration, I wanted to give you a glimpses of how this is going to be from a perspective of what and from an expert perspective of what we will be covering, as well as throughout this entire 3-day experience, what you will be expecting to learn from all of these different experts coming from all around the globe and discussing about multi-omics approaches and next generation and high throughput approaches. So one of the very important question is why we are doing this, why this workshop is important, and why these topics have been covered by a variety of experts. So to simply say this multi-omics and omics data and healthcare data is now being taken care of. Like, people are now concerned about this data. Hospitals all over the world are generating this data, which is being used for clinical outcomes and all of those things from drug discovery. So being a biologist and being a life science student, it's kind of very important for us to learn about this, that what is going on with this genetic data, what is their cause and effect. Because when we discuss about this genetic data, it's just not about the human beings, but also the microbiome and microbes, which is present all around us, and also the environment that plays a role. So we're talking about different types of information, and that's why it is a multi-omics in nature, because there is multiple types of omics data types that explains this nature of and specific system. So I think that is very important, and from that perspective, this workshop is very important for anyone to get an overview, understand, and then take decisions about, may that be a diagnosis, may that be prognosis. There are several applications that we will be discussing throughout this workshop. I have a very short presentation. Quickly, I'll share that. That will give the glimpses of what we will be doing. As we are also collaborative partners with MT University, whom we are working together on omics logic and multi-omics training. So I thought the educational aspect of this is quite important. And that's why I wanted to share some of the important resources that would be relevant for everyone to learn. So just a second, please. I have so many slides opened. So as you all know, as you all might know and might not know, so there's a brief introduction. We are a technical spin-off from the University of Haifa, Tauber Bioinformatics Research Institute, with the help of Dr. Alfred Tauber, Dr. Leonard Brodsky, and all the other co-founders. We've been able to work with thousands of students spanning across India, Africa, and the US. And our team is present in several of these locations, in several of these places, to help the university and especially the faculty. So how we are doing this is through the TBioInfo platform. The TBioInfo platform is developed for anyone who is interested to learn about and apply this sort of data to the research, to different types of application. So it's a multi-omics integration approach that we are taking, where we are integrating different types of data and drawing insights and understanding how they are interconnected for us from a system biology perspective. So in this process, you will learn about different types of omics data, obviously gene expression, and then you learn about mutational data, and then how you how one can make sense of out of all of this, because one of the biggest challenge in multi-omics integration is again the how the different omics data communicate, and what are the connections that we need to find out from this massive amount of information. So that's where T-Banfo platform is an amazing place to be, because it integrates it and gives us really clear insights of where the biology, where the biologist should be looking at further to to understand the insights that are coming from this sort of complex data. So a lot of its application is in pharma, but a lot of its application comes to real patients when we are talking about these panels that are now being used in especially hospitals, many hospitals in India also. So there's a huge application that is ongoing, but still there is a lot of awareness which is still to be there, because there's um, a lot of places where bioinformatics and the application of such data is not known, and the outcomes for the patient's welfare is also not known for the same factors. So the T-Bioinfo platform uses these different sections. So from next generation sequencing to epigenetic to metagenomic, all types of data. And then we have specializations like virology, cancer biology, and different types of analysis for metagenomic sequences, single cell, and then machine learning. So integration of this sort of data requires you to have maybe a common platform for such a comprehensive analysis gives us a clear picture. So most of the users who have used the platform for research perspective as well as for education has shown a really good level of good feedback from out of all of this. So one of the important aspect of doing this is to understand that what kind of data you can integrate and what kind of questions you can answer through that. So that's why this educational program, and as I was telling you that we have some really experienced researchers, scientists going through this entire pathways, completing projects with us in collaboration, utilizing these sort of pipelines where we have made sure that it is easy to understand from an learner point of view, because many of you are learning for the first time by joining the workshop. So how to learn about these different sections, what they do. So there's a lot of visuals. A great graphical user interface always attracts a lot of students or a lot of participants or users, because it's kind of important to make things easier. Because at this level, when we are talking about multi-omics integration, when we are talking about big data, we are talking about dimensionality reduction, we are talking about very high-end statistics. So one of the important thing is to understand the basics very well. So after you learn about the basics, it's kind of important to know what are the different things that you can do from this. So this graphical interface actually helps a lot in this. And we have an upcoming session from Tauber Bioinformatics Research Center, where Dr. Julia and Dr. Elizaveta will be discussing about these sections, where you will be able to learn about these different types of data analysis and the outcomes that one can get. So this was about the platform and about the different sections and the opportunities that it brings to everyone to run their own analysis, to do this research through this platform, and to also generate insights, meaningful insights for biological interpretation, or for going to the clinic or giving that report to the doctor. So all of that integration help is usually through this different complex algorithms that we use, which you might be learning about and hearing about a lot: machine learning, deep learning. So how to make use of these, where this is applied, what kind of data you need to run this sort of analysis, and if the data that you're running is also meaningful for the experiment or not. So there is a lot of ambiguity, there is a lot of confusion, there is a lot of guidance which sometimes is required in this manner, which is done through the platform's own capabilities. Then connecting it with the structure biology. And then I want to quickly tell you more about the how we are doing this in terms of the training with students. And thanks to all the faculty who have contributed, and all the colleagues from our company as well as from MIT University, that led to this multi-omics nature of doing completing research through this different training sections that we have developed through the Omics Logic Learn and as well as TBA Info platform. So amalgamation of coursework along with the practical hands-on has given this opportunity to several of the students to be able to work on multi-omics projects. And we saw that there's a trend in multi-omics project that students from the first year batch were talking about, multi-omics techniques such as transcriptomics, genomics, and the clear indications that people were aware of is mostly cancer and those diseases. But as this awareness is growing, this could be seen from the data perspective as well, because now the projects are infectious diseases, neuroscience, precision medicine, different types of cancer, on different educational backgrounds. So this was something that we noticed while going through the data, that the understanding and the awareness of multi-omics approaches and um, the kind of outcomes that people are expecting with these AI-driven technologies. Like if you find out, there are like at least 20 AI-driven technologies and startups just started in the last 2 months who have clear insights in what they want to do, and hence they are working on these several areas, which are now also seen in the students, that students are learning about it. So these different students worked on this, and through the partnership program, we've been able to do this. And obviously, with the help of all the founders, co-founders from both the partnering institutions and the Top Bioinformatics Research Center, we've been able to come up with a translational research to be able to do translational research while doing your education. So this is the biggest announcement that we will have during this workshop, that the microbiome and health challenge. So many of us are working and we are looking at the microbiome, how does the microbiome affects our body in different aspects. So what are the different ways to convert this idea into a translational project or a translational research or a company or a startup that Dr. Raval was talking about. So this is one of the collaborative work that we are doing, that we will be showcasing, and with all the industry partners that are collaborators who are participating, and the students are participating. The idea is to understand and find these solutions. So here we discussed about multi-omics integration. We discussed about different types of omics technology. But what is the translational aspect of all of this? How do we be able to come up with ideas that goes to the market, and we kind of make a sense out of it, a business sense out of it? And that's the hackathon is all about. It is focused on microbiome and health and wellness. I'll just not take much time, but the idea is to have the conceptual business model, and the business model is something that students from biotechnology and life sciences background does not learn much about, but there are so many entrepreneurs and ideas that they have could be utilized. So a combination of life science students and a business student and a computer science is to work on a hackathon and develop these different propositions through MVP presentations, sequencing, understanding platforms, and then developing a user interface. So these are some of the dates that we will have, and the work that will be done in is to use multi-omics integration, use multi-omics and other types of data for obesity and nutrition, for innovation and beauty products, pregnancy, and femtech. So this is what is something that is coming up in our presentations, and also opportunities for students to learn about and experience this, and to be able to go through the resources and the training, and then able to come up with a translational aspect, a startup, or investors kind of opportunity to go to the investors. So that's the application of multi-omics from the beginner level to a translational level. That will be covered. Some of the glimpses will be covered in this workshop, and I think everyone who's joining will have an exciting time. And again, we will be extending this, and our collaboration is to work with all of you and help you all who are joining us. So we'll be looking forward to all of that during this event and the upcoming events. Thank you so much.
Thank you so much, sir. Now, I would like to humbly invite Professor Sarat Janga to share the keynote address. Professor Sarat Janga is an Associate Professor, Bioinformatics, Data Science, Indiana University. Professor Janga pursued his PhD in Molecular and Systems Biology from University of Cambridge and carried out his postdoctoral research at the University of Illinois at Urbana-Champaign. His research integrates computational and experimental multi-omics approaches to study complex and heterogeneous data sources in biomedical sciences, with the goal of understanding how the regulation, structure, and dynamics of the biological systems shape the phenotypic landscape of an organism and its relevance to disease conditions. We are delighted to have you here with us today, sir. I now request Professor Sarat Janga to kindly address the audience.
Thank you, Sudipti, for the
Good
kind
evening,
introduction.
professors.
Let me see if I can share the screen. Can you see that all right?
Yes, it is.
Perfect. Thank you. Thank you all colleagues at Amity University. It's a distinct and great pleasure to be joining virtually this conference. So I'm Sarat Janga. I'm an associate professor of bioinformatics and data science from the Department of Biohealth Informatics at Indiana University School of Informatics and Computing. I started here in 2012. I want to give few words before we get into the scientific journey of what we do in my lab. This may be of interest for young investigators who are aspiring to be future bioinformaticians or biomedical informaticians. So I did not think I would be in bioinformatics. I started as a bioengineer, biochemical engineer, so to say. Most of the people who take a biochemical engineering are unlikely to get into genomics or imaging or any of these. That's although. So I started as a unconventional kind of a engineer converted into genomic scientist and then into informatician. Now we do both experimental and computational work in biomedical settings. And we also look at health information or medical imaging kind of data, which I will try to touch upon if we have time today. So the work in my lab is supported by a broad range of funding agencies in the US, from the National Institutes of Health. There are sub-institutes of NIH such as NIGMS, National Eye Institute, National Institute of Allergy and Infectious Sciences, National Library of Medicine, several of these fund. In addition, National Science Foundation supports. We're also fortunate to get support from Eli Lilly and Company, which is one of the first companies to have come up with a drug for diabetes. And they're just around the corner at Indianapolis. So we are fortunate for them to have been supporting our work on protein RNA interaction mapping in liver diseases. We are also lucky to have some support from Indo-US Virtual Networks from IUSSTF. This is one of the recent collaborations during COVID that we established with IGIB to build on visualization for dashboards of COVID screening. I won't be having time to talk about several of these scientific outputs that talented students in the lab have been producing. We'll be focusing on couple of things. So, but before we get into the more specific scientific aspects, let me quickly give a detour on what we do in the lab. So my lab started with mostly computational and then emerged into computational experimental approaches for studying how the structure, dynamics, and evolution of post-transcriptional networks shape. Now, when we talk about post-transcriptional network, broad range of networks are going to come into picture. We have been specifically interested or largely interested into how proteins interact with RNA for several years now. And although we have done some work on microRNAs and other kinds of regulators, but mostly how RNA binding proteins interact with RNA and their interactions, their dynamics, and how they change in disease. That has been a good chunk of work. We have also been interested in how these interactions mean anything functioning to the cell. So when you map these high throughput data sets, one of my previous colleagues has been talking about large amounts of data generated from multi-omics. Now, when you have these interactions or cellular interactions, cell is a very crowded environment. And you don't know how many of these interactions actually mean anything functionally. So we have been interested in perturbing these interactions, such as protein RNA interactions, to see using CRISPR-Cas9 screens to see what are the functional consequences. And these generate a lot of data and often with noise. So we have been interested in using computational methods on how we can use this data for functional understanding. The third area of genomics, which has been very exciting, and we started working since around 2016, 2017, is use of nanopore sequencing. So traditionally, even folks who work on nanopore sequencing, they have been most interested in sequencing DNA. But we have been very interested in using direct RNA sequencing of RNA for the purpose of identifying RNA modifications and developing algorithms for mapping these modifications as well as mapping structure. So the bulk of what I will be talking today will be on using this fourth, or some people refer to as the third generation sequencing technology, for sequencing RNA molecules directly and trying to map those modifications using sophisticated state-of-the-art machine and deep learning methods, and how such kind of frameworks can be used to map structures at a single molecule resolution. So what kind of stuff we have been doing? We have been in the lab. On the left top, you can see a simple data integration from various different sources and building of networks, and they're changing the dynamics of these interaction networks in various conditions. So that is one of the typical non-experimental frameworks that we use in the lab for data integration from various sources and studying the structure and changes. Or we have also been using experimental methods, such as you can see in the bottom left, a visualization of a nanopore sequencing reader, where a single pore is sequencing a double-stranded DNA, but one strand is going through the pore, other is getting zipped out, and there is a current that is getting generated as a result of that sequencing. And that data is of interest because we want to see what are the changes in this molecule, right? I'll talk more about this in a little while. In the middle panel, you see variations on how a modified and unmodified RNA molecule will look like on a current, and in its various forms of visualization is shown in the middle panel. On the rightmost on this slide, you see a various set of databases and resources that we have developed. For instance, ReadDB is one of the first databases for RNA binding proteins, and how their expression and how they are associated with various diseases in humans are put together. So that database is very sought after by experimentalists interested in studying any of the 1,500 odd RNA binding proteins in the human genome, experimentally known. And diffhunter is a tool for differential binding analysis using RNA binding protein RNA interactions. Express is a database for expression profiling of genes in the retinal diseases. iSplicer, as the name suggests, it shows splicing events for genes in the eye. Lantern is a database of used based on natural language processing for knowing the annotations of long non-coding RNAs. For a long time, long non-coding RNAs did not have a specified resource such as gene cards. So we have been interested in curating literature using semi-automated methods, natural language processing methods, for annotating long non-coding RNAs, along with their expression in other sources. And that resulted in a database called Lantern. Satan is a tool for doing functional enrichment analysis for RNA binding proteins. Specifically, XServe is a database for studying the importance of exons contribution in various cancer types. So we routinely hear resources for studying the gene's importance in cancer. We're using survival analysis, but we have come up with a resource called XServe, which enables users to study whether an exon is important for the survival of a cancer patient. And using a lot of cancer genome atlas data sets. Peak is a resource. We haven't yet published it, but it is one of the first tools for integrating the binding sites of RNA binding proteins together with evolution of the binding site across species to see whether we can identify functionally interesting binding sites. The last of these panels here is where we will be talking in more depth on using a deep learning architecture for predicting RNA modifications from nanopore RNA, direct RNA sequencing data. So what I thought I will do today is talk about two different or related frameworks where we have used single molecule sequencing from nanopore sequencing technology to map RNA modifications and their structures. So let's briefly go back to the basics before I jump into what we do in the lab and how our experimental and computational efforts are integrated in a broader context. Now, according to the central dogma of molecular biology, DNA gives rise to RNA through the process of transcription. And for a long time, and several groups are interested in how the polymerase, how transcription factors dictate or control the conversion of DNA to RNA at specific times and spaces within cellular contexts. Now, we also know that RNA gives rise to protein through the process of translation. Now, this process is heavily controlled by post-transcription regulators, among many, such as microRNAs. We also have RNA binding proteins. And traditionally, RNA binding proteins for quite some time have been thought to be predominantly involved in translation or to some extent in splicing. Now we know that as soon as genes are transcribed, RNA binding proteins are involved in the localization of RNA, as you can see in the bottom panel, from the transport of RNA from the nucleus to the cytoplasm to different subcellular compartments, their stability through poly-A binders to the 3', and their movement from within the cell to different localizations, their modification, editing, and so on and so forth. So increasing number of post-transcription processes are now attributed to the because of the RNA binding proteins. And what was also thought previously and has been changing in the recent years is each of the steps have been thought to be controlled by a specific group of RNA binding proteins. Well, now we know that several of the steps are could be controlled by one or few proteins. The same protein could be involved in localization, could be involved in translation, and so on and so forth. So the integration of this process can be very tight, and a few proteins can be involved in the control of the metabolism of RNA from end to end. And that has resulted in a very complex network of RNA binding protein RNA interaction networks within cells, which vary from cell type to cell type. And they could also be controlled by protein-protein interactions because RNA binding proteins are known to be significantly unstructured, and their non-RNA binding regions can interact with proteins and spawn complex networks of interactions. So what have we been interested in this context, right? So one area which is very simple or thought to be simple for a long time is RNA is comprised of four major bases. RNA is comprised of four major bases, uracil, cytosine, adenine, and guanine. Now, for instance, in the 70s and 80s, in ribosomal RNA, we have noticed that an adenosine can be modified into N6-methyl adenosine, or adenosine can also be modified into N1-methyl adenosine. You can see that schematic in C and D panels here. Or one could also notice that adenosine can be converted into inosine. For instance, simple flipping of a amide group into a double bond results in adenosine base to be converted into inosine base. Or uridine can be converted into pseudouridine because of flipping of double bond from one location to the other, as you can see in the panel B here. We also now know that cytosine can be methylated in the 5' location, and that results in 5-methylcytidine or 5-hydroxymethylcytidine. And these sort of modifications, we now know that there are about 160 types of these modifications, which we and others have summarized very extensively in reviews. So of these, few modifications such as M6A, A to adenosine to inosine, pseudouridine, and 5-methylcytosine are thought to be very abundant, not just in ribosomal or other non-coding RNAs, but also messenger RNAs. Now, previously, they were thought to be mostly occurring in non-coding, but now we know that protein coding genes have several of these modifications. So that aspect has triggered a lot of enthusiasm to learn why these modifications are important. So let me tell you our perspective of this, and there are other aspects of how and why these modifications could be interesting in the next few slides before we get into the technology viewpoint. If you take an mRNA molecule, what we now understand is an mRNA molecule can have one or more modifications, as you can see in the top part of the schematic. Now, these modifications can occur anywhere in theory along the length of the RNA, and can occur multiple times, and can occur proximal or distal depending on the context. Now, this mRNA molecule is also structured, so we have to keep that in mind. Although I have simplistically shown that this molecule doesn't have, is looking at a flat. Now, the modifications that I've shown on this slide can be broadly classified into two types. there are those base editing modifications, which such as adenosine to inosine or cytidine to uridine, which result in a complete base flipping. And hence, when you perform Illumina sequencing, these bases are read as a different base, okay? Because in Illumina sequencing, you're essentially doing a cDNA sequencing, and hence, the reverse transcriptase are likely to read, because of the base flipping, they would read them as different bases. Or for the most part, these modifications are not essentially base modifications and are simply chemical alterations. Examples of these simple chemical alterations could be M1A, pseudoiridine, for instance, or 5-methylcytidine, or even 2'-oxygen methylation. For instance, on the right side, to the very right, you see 2'-methylation, which is a simple methyl group addition to the backbone of this molecule. These sort of backbone modifications can appear simple, but if you add a heavy, bulky, backbone, which is a charged one, that could result in significant alteration of the potential local structure. So there are a number of these naturally occurring modifications that have been detected, but it's also possible when you expose cells to distinct chemical reagents, you could also have modifications happening in unnatural means. And understanding and mapping these naturally occurring or unnatural modifications can have distinct advantages, as you will see in the next set of slides. So before we again get into the technology viewpoint, I wanted to mention one crucial observation that has been made and has been increasingly appreciated for the importance of the notion of epitranscriptomic code. So several of you might have heard about the notion that epigenome or epigenomic code. So a few years ago, it was observed that when an mRNA molecule, which has an adenosine modification, as you can see in the schematic here, it can be modified into methyl adenosine with the help of what are called as writer proteins. Writer proteins are the proteins which are again RNA binding proteins, but are essentially enzymes and have the ability to convert an adenosine into methyl adenosine, right? In the cell, things don't happen by magic. Adenosine, if it has to be converted to methyl adenosine, it has to be done with an enzyme. And these enzymes, which are essentially proteins such as WTAF, metal-14, metal-3, they have the ability to convert adenosine into methyl adenosine. Now, what was observed in around 2015-16, around those years, is that methyl adenosine can be reversibly converted into adenosine. And this can be done by what are called as eraser group of proteins such as FTO and ALK-BH5. This reversible nature immediately triggered the notion that the RNA modifications has a possibility of getting reversed in the cell over a time point or a period of time. And so it gives a lot of plasticity to RNA molecules. Now, what kind of plasticity could occur because of these changes? So for instance, when adenosine is methylated, the methylated RNA can be read by a different class of proteins potentially, which could result in, for instance, differential binding outcomes of such as translation. Because of differential binding, it could result in changes in stability or altered splicing and nuclear export. So I'm just giving you a high-level picture, but you can imagine the endless possibilities on how the modification can result in altered outcomes for the fate of an RNA molecule. Now, this is the understanding for one type of modification. Now, you can imagine what different RNA modifications and their potential reversible nature and their combination of these modifications could contribute to the cellular phenotypic landscape. So that was more like an open note for all of us to think about the importance of these modifications and what they could contribute. Now, let me quickly jump into a framework that we built in the recent years for mapping RNA modifications. So what we have been interested in is, well, there are a lot of different methods for mapping modifications. Often these are done using antibody-based methods, or sometimes for cytosine methylation, you end up using bisulfite sequencing. But the end product of all these current methods is using short-read Illumina-based techniques. Now, what are the challenges with short-read Illumina-based methods? Well, you could not get the phasing of these modifications. When there are multiple modifications, you don't know whether they happen on the same transcript or a different transcript. And you also do not have information on the larger context of modifications which occur in introns and exons, whether they're, so there are some of these challenges. And you will have to run each time you're interested in a modification. You have to find an antibody for M5C, you have to find an antibody for M6A, and so on and so forth. So each time you want to map a modification, it's a different experiment. So we wanted to circumvent these challenges and see if there is a unifying technology where you would sequence once and you would be able to map several modifications at the same time. This is where we have been excited about nanopore direct RNA sequencing. And what you're seeing on here on this animated image is a single molecule of DNA, which is zipping through this nanopore. And each time the molecule moves through this, you're getting a characteristic disruption of current because of this movement of this molecule. And at any given point in this pore that you're seeing, there are about 5 nucleotides that are sitting in the pore, and the current corresponds to those 5 nucleotides. In a typical nanopore sequencer, there are about 1,600 to 2,000 such pores which are generating such current signals. And these current signals are getting converted into the nucleotide bases, and those are given as outputs of the sequencing data. Now, as you can imagine, because of the massive amounts of data that is getting generated, the processing of these current signatures into nucleotide bases is done using GPUs as opposed to CPUs on these nanopore sequencing machines, which are end-to-end integrations of these nanopore device together with the computing hardware that is associated. So we have been excited and interested in using direct RNA sequencing. A direct RNA sequencing workflow typically looks as follows. You take a full-length RNA molecule from cells, lysed. You can take any cell lines, bacterial cells, any of them. But if you're looking at bacteria, you'll have to attach a poly-A to the RNA. And you would use primer annealing and ligation. And there is an optional reverse transcription step to stabilize the RNA. This is optional, but it is recommended because if you don't stabilize using reverse transcription, the 3' poly-A could form structures and could potentially block the nanopores. So it's often recommended to do a partial reverse transcription step. And you would then attach adapters. The whole process takes about a couple of hours after the full-length RNA is isolated from cell population. And we have done it a number of times in my lab. And this is how a typical device for nanopore Minion looks like. This is about the size of a USB drive or large USB drive. Now you can also have these devices are connected to hardware computing display, your typical laptop. But Nanopore also provides iPad-like devices which integrate the sequencing module with a display module, so that you don't even have to buy a computer. It's end-to-end integrated. So we have used these devices, these pocket devices, or field specific. You can use them on the fields for sequencing COVID and so on. This is the context in which I was mentioning. We have been collaborating with IGIB folks in New Delhi to see whether we can develop on-field sequencing of COVID genomes. So we have used it in this context for sequencing cell lines, for instance, cervical, liver, myeloid leukemia cell lines, as well as mouse primary tissue samples. There are some challenges associated with primary tissue samples that can be a topic of a different discussion. So what is of interest today is I'll just mention about a couple of data sets that we have used in this study. And these are some of the early data sets that we have generated in my lab. You get not as many read counts as you would see in an Illumina machine, but these throughputs have increased to about a couple of million nowadays, as opposed to 700-800K that we have generated a few years ago. This is a histogram of the read length distributions. X-axis shows the read length, and y-axis shows the number of reads that of a particular length. So what we should be noting here is, as long as the length of, what you see is what you get. So if you're seeing that an mRNA molecule is 4,000 bases long, you would see it. I've truncated these plots to 4,000, but we have seen that there are RNA molecules which can be as long as 10,000 or even 15,000, 20,000 nucleotides long, and you would get a read as long as the length. So also of an observation that can be made here is that in liver cells, the liver carcinoma cell line, you see that the read length distribution is slightly tilted towards longer, while in the leukemic cell line, the read length distribution is a bit towards the shorter end. So generally, it appears so that the mRNA molecules are a bit longer in one cell line to the other. So that was some quick observation on that particular data set. So why is this technology of interest? Let me actually explain quickly about the gist of why this is exciting. Every time an RNA molecule, I've shown this visualization of nanopore on the left for the DNA, but the RNA is essentially looking very similar, except that it doesn't look double-stranded. It's a single-stranded molecule zipping through the nanopore, and you would get a current. At any given point, there are 5 nucleotides, and you can see a fimer, how the signal looks like a fimer for a 5 nucleotide window. Now, on the top right, you can see that there is a molecule of RNA whose current is being shown as on the x-axis is the position, on the y-axis is the current. So there are 2 types of molecules whose current is being plotted here. One is an unmodified RNA molecule, where there is no reported RNA modification. The other molecule, which is not shown, is a M6A modification at a particular position, and we are looking at a position around 215 nucleotides, okay, along the x-axis. You will notice that when an M6A is placed instead of an adenosine on this top right plot, there is a change in current at that particular location. And this is exciting and interesting for because you can use the current signal to predict the modification. And this is what has been interesting to us. And of course, we have developed some visualizations, such as Sequoia is a tool for showing how the current looks for normal and modified. And there are other groups who have developed Tombow and Nanomod, which are other tools for seeing the changes in the current signal at a position. And what we have done is, in my lab, we sequenced HeLa cell line using direct RNA sequencing. And fortunately, HeLa cell line has been very well studied for M5C, which is a cytosine methylation, by several groups using bisulfite sequencing. So we have used those data sets and extracted the current signal. Again, I'm not going into too much of the jargon of the data sets, which I'm happy to talk in a Q&A. You can extract the current signal for fimers at every position. And we were able to notice that about 20,000 signals could be extracted for about 2,500 unique M5C locations, which have been reported using bisulfite sequencing. And because we have matched coordinates for which we know gold standard information of M5C from other sources, such as bisulfite sequencing. And then we have nanopore sequencing telling us that we have current signals for each of these potentially modified and also unmodified cytosine locations. And this forms the foundation for our machine learning and neural network frameworks that we have built. In the next slide, what you'll see here is on the left, we are showing the distribution of current for how a current would look like for unmodified versus a modified. A modified 5-nucleotide signal shows a significantly lower current versus an unmodified signal on it, although there is overlap, which is why our efforts to use standard machine learning did not give us a lot of accuracy for separating these one versus from the other. And you will see in the top right, various machine learning and deep learning frameworks that we have used, and with various data sets. And you may be wondering, what are these data sets corresponding to? So whenever you have an RNA modification, it's not that there will be one RNA that is produced. Any given gene could result in any large number of transcripts. Now, some of these transcripts could be modified, other transcripts may not be modified. This results in a concept of editing efficiency or RNA modification efficiency or extent of RNA modification at a given location. For instance, 0.2 on this plot corresponds to the idea that a given location is about 20% of the times that RNA particular location is modified. while 0.6 corresponds to the notion that 6 on 10 transcripts at that location have been modified at that site. So if you use this fuzzy or less probability modified RNA modifications, you result in generating models which are much more noisy, as opposed to using locations which are highly efficiently modified, because our gold standard data is from Illumina and other kinds of sources using bisulfite sequencing. So there is fuzziness embedded. So as you will notice that as you go for data sets with higher probability, your accuracy is going to bump up. Now we have used very complex RNNs, LSTMs, and 5-layer, multiple layers, and we have so far seen that we reach accuracies of 90% for this problem. But the framework that I'm showing here, we have used it for predicting pseudouridine, we've used it for predicting adenosine to inosine, and a whole range of RNA modifications now. And we have few of these papers published, others are on bioRxiv, waiting for the blessings of the reviewers. So what are the challenges with these kind of machine learning, deep learning frameworks for predicting RNA modifications using direct RNA sequencing? Well, you are at the mercy of the gold standard data generated by complementary technologies such as mass spectrometry, short-read Illumina-based data, because you need some training data. And if there are false positives, they would contribute to your models being inaccurate. The other alternative is using synthetic data. You can design synthetic molecules, embed RNA modifications, and sequence through nanopore, and use that data for developing your training models. But even synthetic molecules, they have their own share of problems. We also have the problem of less signal data. In the early stages of the nanopore sequencing, now things have changed, but early stages, the amount of sequencing data has been limited, but now the cost has come down, so you can generate a lot more data and do more flow cells. And most of these modifications that I mentioned, about 160 modifications, there are no experimental methods, any method, for instance, for mapping any modification. So you don't have any gold standard data to develop these training models for nanopore to be able to detect. So those are our challenges in the field. So let me now jump into another area where we have been using this notion of RNA modifications. And you'll quickly see why this problem it has been exciting for us to jump into. So RNA structure has been studied for a very long time, and structures are important for maintaining the regulation and maintaining, in fact, genome function in a number of contexts. There are a variety of roles associated with RNA structures. For instance, RNA structures dictate the accessibility of RNA binding proteins, access to other RNA molecules for RNA-RNA interactions. The secondary and tertiary structure of RNA molecules also dictate the conversion of RNA to protein, or degradation of RNA, and so on and so forth. So there are a number of different functions that are yet to be studied for RNA structure, but we increasingly understand that structure dictates a function in a number of cellular contexts. So how has structure been studied historically? Historically, structure is done using biophysical methods such as 3D structure, I refer to X-ray crystallography and NMR. Now, these 3D structure mapping techniques, one, they're expensive, they're time-taking, and you can usually study on one or few molecules at a time, and you would take years to resolve the structure. So in the last 10 years or so, the field has been pushing itself more towards a 2D structure determination. Secondary structure determination of RNA has been the most sought after. One, because they are cost effective, they're rapid, and you could generate for a large number of RNA molecules in a cell in the 2D structures. And there are a number of such techniques such as ShapeSeq, DMS, MapSeq, LigerSeq, Paris, and so on and so forth. So the gist of all these methods goes with this holy grail slide that I'm showing here. Given the RNA in a solution which has taken up a shape, a particular secondary structure, what you do in most of these techniques is you would throw a chemical reagent which would access the single-stranded regions. As you can see in panel B, a reagent which would access the single-stranded regions or unstructured regions, and would result in a chemical alteration of the base of that structure. Now, the next step would be using a reverse transcriptase, which would try to read these chemically modified RNAs which are already structured. Now, reverse transcriptases, there are of different classes. Most basic reverse transcriptases, when they see a chemical modification or when they see an RNA modification, they would bounce off that particular nucleotide and would result in fragmented molecules. As you can see, the cDNA is shown in blue, where the reverse transcriptase is falling off every time it hits the RNA modification. Now, these cDNAs are sequenced using Illumina sequencing technology. And what you generate are called as reactivity plots, as you can see in the slide, or plot D. Along the length of the molecule, the higher the reactivity score, the higher the number of reads which are dropping at these RNA modified locations, the more likely that the region is unstructured or single-stranded. That's the idea behind all of these techniques. You would identify a chemical reagent which can target a single-stranded region and contributes to RNA modification, because these chemical reagents cannot access double-stranded regions. Now, because of this differential access to unstructured regions, and reverse transcriptase's inability to cross through these modifications, there will be higher reactivity scores in unstructured regions, and you can use this information for predicting the structure. So let me again summarize. Given a molecule, RNA molecule of interest, you would treat with the reagent which can modify the single-stranded region on the left on this panel, and you would have a control where you are not exposing the RNA molecule or the RNA molecules of interest, and you would perform this series of steps where the end product is Illumina sequencing. Higher the reactivity scores, more the reads in unstructured regions, higher the reactivity scores, and hence that region is single-stranded or unstructured, as I have summarized in this slide. Now, these there are several techniques, but ShapeSec is one of the earliest technique which uses one such reagent for doing this, solving the secondary structures of RNA. And the reagent is called 1M7. So we have asked the question, can we use this 1M7 and treat a molecule, but we end up using nanopore direct RNA sequencing as the end product, as opposed to using Illumina short read? Why is that of interest? One, you could solve the secondary structure at a single molecule resolution. And when you have several isoforms, it is important to deconvolute the structures of isoforms as opposed to individual or trying to condense the structure to one particular gene level. So there are distinct advantages for using single molecule techniques such as nanopore. So our interest has been using Hep C, which is one of the RNAs which has very well studied and is about 350 nucleotides long and has a structure in the 5' as well as along the length. So we performed direct RNA sequencing, but for doing so, we first had to do unmodified and shape treated, the 1M7 treated. But in nanopore sequencing, one of the requirements is to have a polyadenylated molecule. So we had to polyadenylate both the unmodified and modified Hep C virus RNA. And once we perform direct RNA sequencing, you can see in the middle panel here, our anticipation is that because of this molecule, 1M7, adding a bulky moiety to the backbone of the RNA molecules, we would end up seeing a characteristic disruption of current in wherever the modifications happen. But we should not anticipate that to be the case for the unmodified RNA shown on the right. And this differential current and hence differential basis that could be called would be our basis for knowing where there would be structures and where there would not be structures. And we have used a modification of these reactivity scores that can be generated as a result of this framework. And you would anticipate that if nanopore makes a mistake in base calling, this would result in a SNP, as opposed to in the controlled unmodified RNA, where you should not anticipate a SNP. What you're seeing here is on the top along the length of this about 350 nucleotide molecule shown on the x-axis, and the y-axis is showing the various modification probability. Okay, the top part is only showing the difference between treated and control, while the bottom part is showing how the scores, right, the modification scores along the length. And for instance, 0.2 would correspond to the idea that 20% of the time at that location, that base is modified. And you will see that for the most part, things are looking similar between shape and control, but there are subtle changes in few locations. And these changes are because of the shape reagent modifying the single stranded region, and hence the corresponding base calling error, and which is what we are interested in using for predicting the structure. So in short, we have used only reads which are at least aligning 70% to the reference sequence, we call SNPs. We use this modification probability. We generate what are called as reactivity scores, as I mentioned, and then we use this information for a structure constraint solving of the secondary structure. There are various ways you can generate these reactivity scores. You can simply say at a given base, how often in the control data set there is a modification or a base calling error versus how often there is a base calling error in the treated one. Or you can do log x by y, the same kind, and absolute. There are various such reactivity score calculations that can be done. We have noticed that log of x by y, or absolute log x by y is a good metric for computing the reactivity scores for solving the structure. And what you're seeing here on this slide is on the rightmost is the NMR-based structure that EMBL EBI has reported for this molecule. The middle one is Illumina short read based shape-seq resolved structure for this RNA molecule. On the leftmost, or panel A, is the structure solved by our single molecule approach. When we compare the structures for sequence of the structural similarity, we see that there were at least 80% similarity between our structure versus the EMBL reported structure. And you can quickly eyeball that there are several fragments which look different compared to the Illumina-based structure. So in general, what we have tried to establish using this is that you can go for solving long RNA molecules at a single molecule resolution using direct RNA sequencing. Of course, there's a lot of room for development in terms of mapping and using even machine learning for mapping these modifications in the structure in this particular project, which is something that we are working on right now. But the scope is enormous. You can solve complete cell line if you generate enough sequencing depth for several RNA molecules simultaneously, although you also have to face the noise that is accompanying with, yeah. So Priyanka, how much time do you have?
Sir, you can take another 10 minutes.
Okay, that sounds good. So
Yeah.
I just wanted to make sure. So
Yeah.
at this point, I would like to thank the work of several colleagues. This is work supported by Nations of Health and Nations of Science. And most of this experimental work was done by a talented a PhD student, Cosina Mayer, in the lab, in collaboration with Julius Lux, who's a faculty member at Northwestern, and his postdoc, Molly Evans. The computation work was done by Vidur, who is also seen on this picture here. Early work on RNA modification modeling was done by Raja, who was a postdoc, then he's back to India now. And we have now a postdoc from Egypt working on developing machine learning models on this. So I don't take any credit. This is all the work of talented students and postdocs in the lab. So in the next couple of minutes, I want to quickly mention about a different area that we have been working. Glaucoma detection using biomedical images or fundus images of the eye has been of great interest to us. We have been working on a number of eye diseases in this imaging landscape. I know I'm completely shifting gears from omics to biomedical imaging. So let me very quickly showcase some of the work that in this space. So glaucoma is a leading cause for irreversible vision loss and blindness, affecting a large population in the world. And the problem has been that traditionally, when we want to detect this damage to the eye, patients would have to go to the hospital and usually get some sort of testing, and the images are visualized by ophthalmologists. I'm running through the slides just in the interest of time. I just want to mention the machine learning deep learning component which comes into picture, as opposed to talking about the basics of the disease. So this is usually studied using tonometry and gonioscopy, for instance, by the ophthalmologist. So what is the problem here? So this is the optic disc of how your retina would look like if you take a deep high resolution image of it. The interesting components here are the cup, which is marked in here, and the disc. So in glaucoma patients, the simple fact is that the cup to disc ratio, if you take the diameter of the cup and you take the diameter of the disc, the cup to disc ratio changes significantly in glaucoma patients. And this has been routinely done by ophthalmologists by looking and manually, and sometimes even making mistakes, because humans are humans, they could make mistakes. So we have been interested in solving this problem using machine learning, deep learning models by extracting this information from large number of fundus images, both in-house as well as from UK Biobank, which houses about 80,000 images, for instance. So as I said, the simple ratio, vertical length of cup to disc, is of interest in this problem. And what have we been doing? We have been using a lot of public data, performing manual labeling of these regions, performing image contrast, and increasing the number of images by rotating and shifting, and using UNet architecture, which is one of the very widely used deep learning framework for extracting features from images, and computing the cup to disc ratio. Okay, vertical cup to disc ratio, it was computed. So I'll skip through several of these, but very quickly, we mask this using manually. We do a color contrast. For instance, each of the our BGR channels is independently enhanced, and we get, for instance, on the left you see a BGR image before enhancement. On the right you see after enhancement. You can clearly see there is a significant improvement in the quality of the image. We do those enhancements, and then we do rotation and other kinds of augmentations to get several images. We then train using UNet model, and this results in model predictions on how accurately we can capture the cup region and the disc region and corresponding diameter. Okay, and we then compute the ratios. Okay, so this efforts have resulted in very high accuracy models for computing these cup and disc ratios. The numbers shown here is for computing the cup to disc ratios, or exact values. It's not the disease prediction. And we have done it for, again, these are some images of real patients and their regions where we do a step-by-step, extraction of the features. And this is how the distribution of the cup to disc ratios look for a group of a random set of healthy and glaucoma affected. You can see that the numbers are a bit more normally distributed for healthy, but for glaucoma affected patients, the ratios are more towards on the lower end. Below 0.7 is something that we have seen. And this is for a toy data set, and we are expanding to a much larger data set to work on this problem. And we are also trying to integrate medical records in this context. So I'll stop here, and I'm happy to take any questions.
Thank you, sir. So we would actually encourage the participants to share their doubts and their questions on the chat box. And I think, Sarath, sir, there are already a lot of questions for you.
Okay, so do you want me to go over the questions in the chat first and start answering?
I'll take questions one by one, and maybe you can answer.
Sure.
So the first question says, is there any average number of modifications of an RNA molecule can have that you could see across your experiments probably?
I'll first tell the truth. We don't know yet, because as I said, for several modifications, we don't even have the techniques. And for the modifications that we have, one thing that we noticed, I didn't mention too much, but we have a database called Epitomy, epitranscriptome modification database, which houses for about 15 modifications, experimentally reported data sets. What we have noticed is that modifications typically occur in the 5' and 3' end of transcripts. Predominantly, there is a huge bump in modifications. And there is no such thing as an average. It's hard to tell because each gene has multiple transcripts, isoforms, and there could be isoform specific modifications. So it's an unsolved mystery. I'm sorry to bring the bad news, but we don't know the answer.
No problems. The second question says, is this mediated by specific modification enzymes only?
That's a great question. So as I said, nothing in the cell happens by magic, although we try to put on magic and get away every now and then as systems biologists. Well, each modification is done by a class of enzymes. And these enzymes are essentially, as I said, RNA binding proteins, because we give different names for these groups. But for instance, ADAR enzymes cause A to I editing. Or uridine to pseudoridine is done by PAS enzymes, for instance. M6A enzymes is what I've shown on the slides. Metal 14, metal, few of these have been identified. So yes, each modification is done by specific groups of enzymes. And there are groups, research groups, working on identifying new enzymes which are able to do these in both human and other model systems using mass spec and other means. So it's an ongoing journey.
Okay, thank you. The next question says, how balanced are these data sets for training of DL models?
So good question. So I didn't mention it, but we always balance. For instance, I was saying there are 20,000 signals for modified M5C in one of my slides. What we ensure for looking at random cytosine locations in the RNA and make sure that there are at least 20,000 unmodified locations around the cell, because it's easy to get unmodified, much harder to get modified locations. So we always balance. Sometimes we try to do 2 to 3 times the modified signal just to see how the models behave. But yeah, we do balance.
Okay, thank you. Next question says, can we create a CNN network with 4, 6, or 8 layers?
You could, potentially you could. There are ways to do, that's not a problem. But we have seen that CNNs have not been too effective for us in our RNA modification framework. Now we have some models which are not complex. So the other thing I want to mention is we are against fancy models which take forever to run. The reason why I mentioned that is you need base calling for every base, and there is a lot of data when you do sequencing, and you can't have your favorite model which takes 10 hours to run because this needs to be done in real time. Even on a GPU, if your models are complex, they take forever. So we are always favoring, if there are 2 models which give similar accuracy or slight differences, we prefer models which have less overhead on the machine and do much faster run. And that's why we are always going for less expensive overhead models.
Great. The next question says, how does the accuracy vary throughout the run? Is the accuracy stable, or how do you handle it?
So we usually generate loss curves with the epochs to see how the loss curves behave, and this is, I think, in industry standard and standard in the field, that nowadays, if I review a paper for deep machine learning and I don't see loss curves, I'm upset. I think loss curves are very important, and if you see a change in the accuracies as the loss curves are changing, you want to immediately stop your training and go to the previous step where the accuracy, and start reporting it. Yes, okay. That is important. You don't want to have overfitted models, which is typically a problem for deep learning models.
Okay, that's great. I think that's all the questions we have for you, and it was an excellent talk. We thoroughly enjoyed listening to you. Over to you, Sudipti.
Yes, ma'am. We have Shoma ma'am here with us today. So I now request Dr. Shoma Nandi ma'am Professor and Head, Center for Cellular and Molecular Biotechnology, Amity Institute of Biotechnology, Amity University, Noida, to share her thoughts with us. Welcome, ma'am.
Thank you. Hello. A very good evening to everyone. My heartiest congratulations to Dr. Kamal and his team for organizing this workshop. And if I'm not wrong, probably this is one of the first workshop where we have two international collaborators working together with Amity Institute of Biotechnology. Although this is not my area of expertise, still I found the keynote address by Professor Sarath marvelous. Though I need to go through various points to understand and make my views clear. Now, more to hear from all of you in the coming days, and I feel privileged to be present here. All the best to the whole group. Thank you.
Thank you, ma'am. I would now request Professor Kamal sir to please share his remarks.
I think there's some technical problem there. Yeah. So, Professor Sarath, I take this opportunity again to thank you so much for being there with us. I know it's 7 a.m. there, and we've been disturbing you quite early, but it was marvelous and it was an excellent talk that you gave, and I'm sure the participants must be benefiting with it a lot.
Yes, I need to add it because this is not exactly my area, but I just entered and I kept on listening. I mean, it's really nice talk.
Thank you all very much. I'm looking forward to more exciting interactions with colleagues at Amity. Not sure several of you know, but we, as a delegation from Indiana University, we just recently visited Amity, and it was fantastic. We had great experience, and I'm looking forward to more exciting interactions and collaborations moving forward. So yeah, thank you all for inviting and hosting me for the talk. Yeah.
Thank you, sir. Over to you, Sudipti.
So I would now like to invite Dr. Abhishek Sengupta sir, Assistant Professor, Center for Computational Biology and Bioinformatics, Amity Institute of Biotechnology, Amity University, Noida, to please share the vote of thanks. Over to you, sir.
Thank you, Sudipti. Am I audible?
Yes, you are audible.
Yeah. So thank you, Professor Sharad. And with this, we come to an end of a very stimulating and thought-provoking inaugural session of the whole workshop. The session was highly engaging and stimulating. With the advent of digital transformation, plethora of omics and biomedical data is the new healthcare currency. Enormous quantities of data are getting produced from patients using consumer sensors and monitors at home diagnostics and digital therapeutics. Nevertheless, for digital transformation and product innovation in the biotechnology and healthcare sector to flourish and more startups being promoted, it is pertinent to train manpower in accordance with this transformation, and new techniques and approaches must be updated in the curriculum to enable such transforms. I, on behalf of the organizing committee of this skill enhancement workshop on multi-omics data integration and data analytics organized by Center for Computational Biology and Bioinformatics, Amity University, Uttar Pradesh, Noida campus, would like to express my gratitude to our respected founder president, Sir Dr. Ashok K. Chauhan, our dynamic chancellor, Dr. Atul Chauhan, Sir, for being our guiding source of inspiration. I would like to express my gratitude to our motivating Vice Chancellor and Officiating Director, AIB, and the workshop chairperson, Professor Dr. Balvinder Shukla ma'am, for being the catalyst that stimulated us to do our best and standing as our pillar of strength. I would like to take this opportunity to put all my appreciation in the form of words towards our distinguished guest, Dr. Mohit Mazumdar and Professor Sharad Janga, for providing us with their valuable insights, perceptions, and sharing their visions. We look forward to having more such insightful sessions from you in future. I would like to take this opportunity to put all my appreciation in the form of words towards our Deputy Director, Professor Dr. Deepshikha Pandey Katare, Assistant Director, Professor Dr. Seema Bhatnagar, and Professor Dr. Puniti Mathur for their support. I would also like to express my sincere thanks to Professor Kamal Raval, HOD, CCBB, for encouraging us at every step and helping us with all the tiniest of details and guidance at every step of the preparation for this workshop. I would like to thank all the respected center heads, including Professor Shoma ma'am, who is here, and assistant directors, faculty members, and office support who have blessed us with their presence for encouraging us throughout this session. I would also like to place on record my deep appreciation to the workshop program directors, Professor Kamal Raval and Dr. Priyanka Narar, session in charge, Mr. Ankur Chaurasia. I thank all the members of the MIDA organizing team for making this virtual event a great success. I'd also like to appreciate the IT support team and PhD scholars of CCBB for all the necessary arrangements. Last but not the least, I thank all the participants for your attention. Thank you, everyone. Have a pleasant and good night. Thank you.
So I would now request everyone to attempt the quiz, the link for which has been shared in the chat box. Quiz will be hosted at the end of every day, and it is mandatory for the certificate. And we would also request all of you to share your feedback with us. So both the links have been shared in the chat box. So thank you everyone for joining today's session. See you all tomorrow, and make sure you attempt the quiz and fill the feedback form. Thank you.